Ethical implications of DNA technology. Technology and DNA. To the left, you can see the different applications of technology into the field of DNA. There is genetic engineering of plants, which is used in agriculture, genetic screening, which is finding genetic diseases, DNA fingerprinting, cancer treatment, finding the genes that cause some forms of cancer, and using that information to develop medication, genome sequencing, and cloning. Early concerns, recombinant DNA technology. Early concerns about potential dangers associated with recombinant DNA technology focus on the possibility that hazardous new pathogens might be created. What might happen, for instance, if cancer cell genes were transferred into bacteria or viruses? To guard against such rogue microbes, scientists developed a set of guidelines that were adopted as formal government regulations in the US and some other countries. One safety measure is a set of strict laboratory procedures designed to protect researchers from infection by engineered microbes and to prevent the microbes from accidentally leaving the lab. In addition, strains of microorganisms can be used in recombinant DNA experiments are genetically crippled to ensure that they cannot survive outside the lab. Finally, certain obviously dangerous experiments have been banned. Genetically modified organisms, or GM organisms. Today, most public concern about possible hazards centers not on recombinant microbes, but on genetically modified organisms used as food. In common language, a GM organism is one that has acquired by artificial means one or more genes from another species, or even from another variety of the same species. Advocates of a cautious approach toward GM crops fear the transgenic plants might pass their new genes to close relatives in nearby wild areas. We know that lawn and crop grasses, for example, commonly exchange genes with wild relatives via pollen transfer. If crop plants carrying genes from resistance to herbicides, diseases, or insect pests pollinated wild ones, the offspring might become superweeds that are very difficult to control. As for the risks to human health from GM foods, some people fear that the protein products of transgenes might lead to allergic reactions. Although there is some evidence that this could happen, advocates claim that these proteins could be tested in advance to avoid producing ones that cause allergic reactions. Advances in biotechnology have allowed us to obtain complete genome sequences for humans and many other species, providing a vast treasure trove of information about genes. We can ask how certain genes differ from species to species, as well as how genes and ultimately entire genomes have evolved. At the same time, the increasing speed and falling cost of determining the genome sequences of individuals are raising significant ethical questions. Who should have the right to examine someone else's genetic information? How should that information be used? Should a person's genome be a factor in determining eligibility for a job or insurance? Ethical considerations, as well as concerns about potential environmental and health hazards, will likely slow some applications of biotechnology. There is always a danger that too much regulation will stifle basic research and its potential benefits. However, the power of DNA technology and genetic engineering, our ability to profoundly and rapidly alter species that have been evolving for millennia, demands that we proceed with humility and caution.